Thanks so much. Thanks to Eric and Sabera for the invitation and for organizing. And yeah, that last talk where it ended, trying to stitch together multiple patients is very, very relevant to what I'm going to talk about. So I'm excited to be here. So as I warned Eric and Sabera at the outset, there's not going to be any human data in this talk. I've never worked with human data, but I am going to be talking about a general purpose statistical method that may be relevant to this kind of data and this community. So in particular, I'm going to talk about distances on neural representations that satisfy the triangle inequality and capture noise correlation structure. And that's a mouthful, so I want to take a minute or so to break down the three components of this title. The goal of this talk is going to be to construct measures of distance or distance functions between population level neural representations or latent spaces or neural manifolds, if you will. So in other words, what I'm going to describe is a procedure where you hand me two neural recordings, let's say, from two different animals, and I'm going to spit out a number that quantifies their similarity and has certain nice properties. And in particular, there's two nice properties that I think is unique to our approach. The first is that we satisfy a very important property of distances, which is called the triangle inequality. And really quickly, this just means that the direct distance between any two points in the space that you're measuring distances is minimal. So the distance, if I take a direct flight from New York to Los Angeles, the total distance traveled is always less than the distance traveled if I have a layover in Chicago or anywhere else. And that's a really simple and somewhat innocuous property, but it ends up having really, really important consequences, as I hope to show you. And the second kind of unique property of our approach is that we have a way of characterizing differences across animals or differences across networks at the level of trial-by-trial variability. And what this means is that we don't need to average across trials, and we can actually compare different representations at the level of single trial resolution. So trial averaging is a very convenient tool. We do it all the time in my group, but deep down, we always feel a little bit of shame every time we trial average. And I really hope that everybody feels a little bit of shame, just because the noise is a fundamental property, I think, of neural circuits and computation, and it's one that's really understudied, and we have a very small, a very limited understanding of it. Okay, so taking a step back, the overall motivation for working on this set of problems is a recognition that the way we understand complicated systems, perhaps particularly in biology and other natural sciences, is through a comparative lens. So I hopefully don't have to spend too much time convincing you of this. In neuroscience, our understanding of the functional purpose of many brain regions is really built on this comparative approach, comparing across different individuals, particularly across individuals of different species. And you can come up with many, many similar examples of this in our understanding of, for example, gene function is based on comparisons of wild type and knockout, or spontaneous mutations across individuals, perhaps of different species. And of course in behavioral science, and on and on and on. So in neuroscience, I've already mentioned that this has bled into our work a little bit, but I think at a kind of systems level or neural population coding level, this kind of thinking is still nascent and growing within our field. So I'd like to say that we're not the first to do this. There are a select number of, I think, very beautiful works in this area, particularly if you go all the way down to invertebrate systems, where we can have one-to-one identification of neurons across individual animals, and we can compare neurons on a sort of direct level and characterize their variability. So if anybody in the room for some reason hasn't looked into the work of Eve Marder and colleagues, I highly recommend that. But I would say a full understanding of variability in mammalian brains is much more complicated, because we have a less solid understanding of what individual neurons are doing. And really, to study this problem properly, I think we haven't had the right data sets until just recently, and it might even take another 3 or 5 years to really flesh out the experimental approach and technology. So in particular, I want to highlight this data set, which will come up a few slides later, by the International Brain Lab. What you're seeing here are learning curves, behavioral learning curves of roughly 100 mice. They're performing the same two alternative forced choice task. The details of the behavior don't matter too much. The important thing is that we have this really exciting initiative of multiple labs doing the same experiment, recording from the same area in animals doing the same behavior. And what you see from data sets like this is that there's a remarkable amount of behavioral variability across mice. Some mice learn the task really, really quickly, and others learn it very slowly if they do so at all. So the overall point of my talk is kind of to say, wouldn't it be great if we could understand how this animal-to-animal behavioral variability that we often kind of just average away and sweep under the rug, how does that manifest in animal-to-animal variability at the level of neural population geometry or the geometry of latent spaces or neural dynamics, things like this. So again, the goal of this talk is to ask, what is the best way of quantifying similarity? So on this slide, I'm going to tell you exactly what I mean by what we're quantifying the similarity between. I've been using the word neural representations, but you might substitute in there neural manifold if you want. I'm going to start with just the simplest setting, and we can extend and relax this setting pretty easily. But in the simplest setting, we can just treat neural networks as input-output machines, so deterministic functions. If we have two animals or two networks, we're going to label them as two functions, fx and fy. And then we have some set of experimental conditions that we expose both animals to. So I'm going to call these probe inputs, and I'm going to denote them as u1 through um. And this is a very general framework. It applies to many different experimental settings in neuroscience, but I think it can be helpful to maybe ground us in a specific example. So a simple example is think about recording from visual cortex, and you're showing the animal a series of natural images. So we're going to feed in m different natural images into these two neural networks. Again, we can think about this very generally. So we're feeding images into perhaps an artificial deep network and looking at the hidden layer response, or we're doing a high-dimensional recording in visual cortex in a mouse or a non-human primate. So the upshot of all this is that you run your experiment and out comes data like this in a matrix, where each row of this matrix corresponds to the average firing rate of n neurons to your m different stimulus conditions. And of course, everybody at Cosyne should be familiar with the kind of geometric neural firing rate state space perspective. You can think about your data set as essentially being a labeled point cloud in a high-dimensional firing rate space. So each image is going to map onto a different point in this high-dimensional firing rate space. And again, if you like to think of neural manifolds, you can think of these points as being kind of landmarks on the underlying manifold, that if you had an infinitely many inputs to your network, you could sample the manifold perfectly and reconstruct it. We're not going to necessarily assume that. We just have landmarks that are on this underlying low-dimensional or high-dimensional manifold. Okay. And again, there's no temporal dynamics here, but hopefully it should be clear that you could, just with a little bit of massaging this approach, you could extend it to time-varying dynamics if that's what you want to study. Okay, so that is what I mean by a neural representation. And remember, our original question is how can we compare two neural representations across two different neural recordings? And hopefully it should be obvious that this problem is non-trivial. The central issue is that the neurons in each network, unlike in certain invertebrate systems, are randomly sampled and arbitrarily indexed in each individual. So if you just naively subtracted these two matrices and took the root mean squared error, that would be a completely meaningless notion of similarity. So this isn't a trivial problem. I think it's maybe worth pausing here for like 5 seconds to think about, if you haven't seen this before, how would you quantify similarity of two matrices like this? And if you look through the literature, you'll see that people have been confronted with this problem, and they've come up with a very large number of methods. And I would say we haven't yet come to a consensus as a field about what's the best set of approaches, and are there certain cases where you'd want to use one of these methods or another. So this was kind of the genesis of this project, was me looking at all of this and saying, what is the common theme here? Is there an overall framework that we can kind of build? Like a simple idea, just to give you a flavor of the approach, is you could maybe do a linear regression to predict the matrix Y from X, and then use the R squared as a notion of similarity. That's been used in the past. People in the audience may be familiar with representational similarity analysis, or RSA, which is very popular in the fMRI field. In deep learning, people are also interested in comparing hidden layer activations across different networks and trying to reverse engineer or interpret them. The most popular method seems to be centered kernel alignment, or CKA, which is very, very similar to RSA. And then other people have used things like canonical correlations analysis, et cetera, et cetera, et cetera. What I'm going to show you is that all of these, or most of these, can kind of be viewed as special cases of the approach that I talk about, with some modifications around the edges. But not to get lost, for the trees within the forest, or whatever the saying would be, I think all of those methods, work reasonably well if you have a pretty simple setup where you just want to quantify similarity between a pair of networks in isolation. So if you're really just looking to put a number on things, I think this is a relatively easy thing to do, and many of the methods, that I was just showing previously would work reasonably well. Maybe you compare this number that comes out to a shuffle control, and you say, hey, things are more similar than you would expect by chance, and p less than 0.05. That's fine. The much more interesting and difficult problem that I think we're going to be confronted with now, and if not now, 3 years or 5 years from now, is what do we do when we have a large number of networks that we all think are performing a similar task, or maybe have a similar latent space, like, for instance, the International Brain Lab data set that I was showing you. So now you have 100 mice performing the same task, and you want to ask questions like, can I find clusters of mice that have similar neural manifolds, and then map that onto them also having similar solutions? Is there a notion in which I can define the average manifold for a cluster, or an average manifold across the full data set? Can I actually do supervised learning within this setting to predict, will an animal have a good behavior just by looking at its manifold? And can I do dimensionality reduction at the level of the network space? So can I embed all of these networks into a low dimensional space? And that's exactly what the methods that I'm going to show to you are able to do. This is just a teaser of a result that I'll show you later, where we applied this method treating each brain region within the Allen Brain Observatory dataset as a different quote-unquote network or neural representation. And then we get a low-dimensional embedding where 2 points are close to each other if those two brain regions have a similar set of neural representations. OK. So with that motivation aside, let's back up and start from the beginning. We want to come up with a notion of distance between two matrices x and y. And I'm going to argue that there's three things that we want to satisfy. The first is that the distance between x and y should be 0 if I can point to what makes two representations or latent spaces equivalent. The second is symmetry. The distance from x to y should be the same as the distance from y to x. And then the third and trickiest to satisfy is this triangle inequality I was telling you about, which is that the direct distance between x and z is always minimal. So the first question is, why do we care about these three axiomatic definitions? Mathematicians call distance functions that satisfy this a metric space, and why do we care what the mathematicians came up with 100 years ago? The answer is that there's a lot of statistical analyses, like I was mentioning, like clustering, that really only makes sense once you have defined a rigorous metric space. So if you suppose the triangle inequality is violated, you can get these contradictions where you have network y is close to x and z simultaneously, but x and z are far apart from each other. And the left configuration of points would suggest that you should put these three networks into the same cluster, but in contradiction, the right configuration of points tells you that x and z should be put in different clusters. So you run into these kind of paradoxes, if you will. And another important thing that the triangle inequality lets you do is it lets you define a neighborhood around each data point. So let's say each point in this space represents a different network, similar to what I was showing you before. Maybe I can color each point by the performance of that animal on a task. And then if I observe a new animal, I can measure how similar that animal's manifold is to the other animals, and do some sort of prediction based on its neighborhood in this space. And defining that animal's neighborhood only works if you have a rigorous metric space. So that's the kind of flavor of why we care about this. And skipping over this quickly, existing notions of similarity, like using linear regression, don't define metric spaces. In fact, linear regression isn't even a symmetric notion of distance. So that is just a quick cautionary tale that I feel like a public service announcement is necessary. You can ask me questions about this, but I'll move by it kind of quickly. Okay, so just to jump to the punchline, our approach to developing metrics on neural manifolds or neural representations is to borrow ideas from this mathematical field of shape analysis, and these are three books that have really informed my thinking on this problem. Just to give you the brief flavor, you can think about, again, you have a labeled point cloud in a high-dimensional space that you've measured in two different animals. And the issue is that this point cloud is randomly oriented with respect to each other because the neurons are completely shuffled or different across the two animals. So a simple idea is to find the optimal rotation that aligns these manifolds as best as possible and then measure their distance in this aligned space. And if you do this, this is just another illustration of the same idea. We're going to find the optimal rotational alignment, and we're going to compute, for example, the root mean squared error in the aligned space. That's known as the Procrustes distance, and it can be shown that this satisfies the triangle inequality and is also symmetric. But we don't just have to stop there. There's a whole kind of space of metrics that we can develop. For example, we can pre-process our data by whitening the data, the manifold in X and Y, and then doing a rotational alignment. Then we're going to get a different notion of distance out, and it turns out that this notion of distance is really closely related to canonical correlations analysis. So I don't want to get lost in the weeds, but our goal here isn't necessarily to come up with quote-unquote the best metric, but just to give outline like a full space of metrics that all have these rigorous guarantees associated with them and relate them to existing kind of heuristic approaches that people have come up with before. And you can read our paper for the details of that. Okay, and what the outcome of this is, because we've embedded each animal or each neural recording in a metric space, you can think about the space of all possible neural manifolds. And I'll call that as the neural shape space. Again, this is borrowing terminology from this existing field of shape analysis. So the gray surface is a manifold itself, but that manifold describes the set of all possible neural manifolds that you might have achieved from recordings in different animals. And each point on this space corresponds to a different recording that you would have made. And if you try to embed that in a two-dimensional Euclidean space, you can do that by a method called multidimensional scaling, which is well known. You get the picture that I was showing you before. Really quickly, I want to show you some very preliminary results, and we're still actively working on this and hoping to make it better. But I want to show you this analysis applied to the International Brain Lab and show you that we can actually do the prediction task of predicting an animal's behavior using these methods. This is work that's being led by Lyndon Duong, who's a senior grad student at NYU in Aerosim and Chelly's lab. So again, we're looking at this data set from the International Brain Lab. We're looking at the subset of the data where they targeted a neuropixels probe to the same coordinates in every animal. And we're recording mostly in visual cortex because it's a visual decision-making task. So I'm just going to show you a schematic here. Remember, each point here represents a different animal in this data set, so different mice. And each mouse is going to have a psychometric curve on the task that the IBL was performing, which is a two-forced alternative choice paradigm. And the idea is very simple. We're just going to take a mouse, we're going to essentially hold it out and look at its nearest neighbors in neural manifold space or shape space. And then we're going to average the psychometric curves of this neighborhood and see if we can predict that mouse's performance. And when we do that and compare it to a shuffle control where we shuffle the animal labels and positions on the manifold, we do better than chance. And if you look at the individual psychometric curves, they look quite reasonable. And this is actually very astonishing to us because there is a lot of variability in what brain region ends up having the most units that are sorted from. And also the animal's behavior is very, very noisy. So if you look at the black points, which are just the behavioral data or the maximum likelihood fit to the behavioral data, there's very big error bars there. So again, this is a preliminary. This is a week or two old at this point, and we're working on ways to validate this and make it better. But I think it's at least a promising sign that this framework has more room to run with. What time should I stop, Eric?
You've got another
10 minutes?
Yeah.
Okay, great. Okay, I think I can slow down then. I can relax and breathe up here.
10 minutes plus.
10 minutes plus questions. Okay, so I'll still stress out a little bit. So remember, there was like two parts of the talk that I wanted, or 2 points that I wanted to convey in this talk. The first was that satisfying this triangle inequality gives us like access to all these really cool methods like dimensionality reduction at the level of individual animals, and we can do prediction. So that's really cool. But there's a big problem, I think, with everything that I've presented to you so far, and that was, on a very early slide, I said we're going to assume that neurons or neural circuits are deterministic input-output machines. And that actually is a really, really bad assumption for biological networks. So rather than thinking about each stimulus condition being mapped deterministically to a point in a high-dimensional firing rate space, instead I want you to think about a more realistic picture where each stimulus condition maps to a probability distribution over neural responses. And if this is a more realistic depiction of what biological circuits, and by the way, many deep neural networks as well, operate like, how can we measure similarity in the manifold structure in this probabilistic or stochastic case? And in neuroscience, there's a really deep and long history of studying this problem in the context of sensory encoding. So I think this is another really good motivation to work on this problem. So imagine I just have 2 stimulus conditions, stimulus 1 and stimulus 2, and I have isotropic noise around each neuron's firing rate. So here, this is a 2-dimensional firing rate space. The black line is the optimal linear classifier, and it does okay on this task. Now, people realized that if the trial-by-trial noise was, unfortunately oriented in the direction of that optimal linear classifier, your decoding performance is going to be much, much worse. And these are so-called information-limiting noise correlations. In contrast, you might have a situation like this one on the right, where the distance between the average response to stimulus 1 and stimulus 2 is the same, but the noise is actually, as a blessing, oriented in an orthogonal dimension, so your decoding performance is quite good. So these are figures that I took from this really nice and landmark review by the authors here. And there's many, many other works dating back to the mid-90s where people were interested in this problem, and it's still actively being studied today. And the point is, if you just looked at the trial average response, you would entirely miss all of this really important structure that has implications for how do we expect animals to actually behave on the task. Okay, so we wondered how we were going to compare stochastic representations, and this was joint work, again, led by Linden Duong with co-first author Jingyang Zhao and the other trainees listed here. The idea is going to be exactly the same, that we're going to try to find the optimal rotation that aligns these two labeled probability distribution clouds into a common space, and then we're going to measure the distance between these probability distributions. And without getting caught in the details too much, there's 2 nice forms of distance between probability distribution. One's known as the Wasserstein distance, or Earth movers distance, and another is the energy distance, which is essentially the same as the Wasserstein distance, but using a maximum entropy or maximally random transport plan. And what's nice about both of these forms of distances is in the limit, as your trial-by-trial variability goes to 0, your probability distributions are going to converge to delta functions, and then the distance between those delta functions is going to become the Euclidean distance. So in the limit that there's no trial-to-trial variability, we recover the original shape metrics framework that I was telling you about in the beginning of this talk. So this is a true generalization of the approach to stochastic data. Okay, very quickly I'm going to show you a demo on simulated data, and then a couple of quick teasers on real data, and then I'll conclude. So this is a simulated data set where I'm showing you 16 really toy networks where each color corresponds to a different stimulus condition, and you can see that the mean response to each condition is the same across all these networks. So if we just looked at the trial average activity, all of these networks are the same manifold structure. But you can see what I've done is vary the noise correlation structure and also vary the scale of the noise as you go down. So as you go down this plot, the scale of the noise is increasing. So what we're going to do is take samples from these network. We're going to compute a pairwise distance matrix. So I forget exactly how many networks we had here, something like 96. So we're going to have a 96 by 96 distance matrix where element ij is the distance between network I and network j. Because this satisfies the triangle inequality, all these distance comparisons are self-consistent in some rigorous mathematical sense. And then we can use multidimensional scaling to visualize all the networks in a low-dimensional space. And we get out a two-dimensional grid of networks, which corresponds exactly to this two-dimensional grid on the left. So we back out the differences across networks in terms of the scale of their noise as well as the noise correlation structure. And if you do this in any other way, if you use any existing method, by construction, they're all never going to look at this structure because they're only operating at the level of the trial average activity. Okay, really quickly, one thing that we're interested in is applying this to deep neural networks. So Sabera was mentioning, for example, latent or deep variational autoencoders as a way of extracting interpretable latents from complicated data sets. We looked at a simple synthetic data set that was released by Google with thousands and thousands of these autoencoder networks trained with different objective functions and different architectures. And what you see is that each choice of architecture and each choice of objective function gives you a slightly different latent representation. And we wanted to leverage our tool to compare all these latent representations. And importantly, the variational autoencoder adds noise to the hidden layer. So this is a really good use of the stochastic extension of our method. Without going into the details too much, we see that there are differences, so you get different latent spaces depending on the modeling choices you make, which is maybe not too surprising in the abstract, but I think this is also a cautionary tale for everybody because typically what happens is people just train a variational autoencoder, and then the first one that kind of works, we're just going to stick with that and do all of our analysis downstream of that. But when you do a really systematic search over the space of different deep networks, you get different results, and I think it's an open question of whether the scientific conclusions are going to differ. Really quickly, we're also applying this to noise correlation analysis in neural data sets as well. That work is just getting started now, but stay tuned for that. So really quickly, let me just summarize what I've said and take a couple questions. So I think this approach of comparative systems neuroscience is a real emerging opportunity for us, and I'm very excited to see what comes of it. I think that when, as soon as we start grappling with data sets with many, many animals, we want to think about developing, rigorous approaches that have theoretical guarantees. I think the triangle inequality is something that's really important and has been missing so far, and we were able to address that. Again, we're not trying to come up with quote unquote the best way to do this, but we're trying to explore the space and give you a recipe book for constructing a metric that satisfies the triangle inequality. And we also have a really nice way of thinking about this in terms of the geometry of neural manifolds, and this extends very nicely to the case of having stochastic manifolds or stochastic representations. So with that, I think I'll just thank everybody who was involved with this work and take questions.